Hello friends, here is a reasoning question related to the digestive tract. H2 blockers like ranitidine, they block more than 90% of the acid secretion. Explain why. So let's explain. But before that, first you need to understand the angle of that question. What exactly is being asked? Uh, if we talk about the acid secretion, it is by the parietal cell in the wall of the stomach. Now this parietal cell has got three stimuli which act on the parietal cell and cause HCl secretion. Neurocrine stimulus, that is nerve, vagus nerve, releasing acetylcholine, and it acts via the M3 muscarinic receptors. Gastrin is the endocrine stimulus which has its receptor on the parietal cell, CCK2 receptor. So gastrin acting via that receptor can stimulate the HCl secretion. And the third one, which is in question here, the ECL cell, enterochromaffin-like cell. This is a paracrine stimulus because it's just adjacent to the parietal cell. It secretes histamine. The ECL cell secretes histamine, and histamine acting via the H2 receptor, and H2 receptor is located on the parietal cell, of course. So histamine acting via the H2 receptor, it stimulates the gastric acid secretion. And H2 blockers are the drugs which block this histamine action on the H2 receptor on the parietal cell, and they inhibit the HCl secretion. Now the question is, it is only one stimulus that is being blocked. H2 blocker, ranitidine, it is blocking only one stimulus. There are still two other stimuli available that can stimulate the parietal cell, the acetylcholine, vagus and acetylcholine, and the gastrin. Then how is it that just one stimulus getting blocked and 90%, more than 90% HCl secretion gets blocked? How is that? That's the angle of the question. So let's understand this. Look, gastrin and vagus, that is acetylcholine, they can act independently on the parietal cell via their own receptors. They can cause gastric acid secretion. However, their more potent action is via histamine. Gastrin and the vagus, they cause histamine secretion by the ECL cell, and then that histamine acts via H2 receptor to cause HCl secretion. So the point is, histamine plays a pivotal role in the gastric acid secretion. Not only histamine acts directly via H2 receptors, but the other two stimuli are also acting via histamine. The other two stimuli are also, their main action is via release of histamine from the ECL cell, which will act on the histamine H2 receptor to cause HCl secretion. Mind you, the other two stimuli, that is vagus and acetylcholine and the gastrin, they have their independent actions on the parietal cell, but their more potent action is via causing release of histamine from the ECL cell, and that histamine causes HCl secretion. So that is where our answer lies. We have written it here: gastrin acting via CCK2 receptor can independently cause HCl secretion. Vagus and acetylcholine via M3 muscarinic receptor on the parietal cell, it can cause HCl secretion independently, and then the histamine. And as we have mentioned, gastrin and acetylcholine act via action on the ECL cell. So histamine has a pivotal role. Now, so when you give H2 blockers, the action of histamine via those H2 receptors, that action is blocked. And that means the other two stimuli, their actions are also getting blocked when you give H2 blocker, because the other two stimuli are also acting via histamine release and histamine acting on the H2 receptor. So when you give H2 blocker, their actions are blocked. Additionally, now that's also an additionally important point, Basically, the ECL cell, when it releases histamine, and histamine acts via H2 receptor to cause acid secretion, it acts via increasing the cyclic AMP levels inside the parietal cell. Histamine action on the H2 receptor causes increased cyclic AMP levels, and that is how the HCl secretion is increased. So when you give H2 receptor blocker, H2 blocker, what really happens is it decreases, those drugs, they decrease the cyclic AMP levels inside the parietal cell, and actions of gastrin and acetylcholine are weakened because of a decrease in cyclic AMP. Because if H2 blockers are decreasing the cyclic AMP levels inside the parietal cells, that means there would be no protein kinases. There is decreased action of protein kinases through which those other two stimuli were exerting their actions. So decreased cyclic AMP, decreased protein kinases when H2 blockers are given. They block the action of histamine on the H2 receptors, and that will weaken the action of other two stimuli as well. And therefore, H2 blockers, although they are just blocking one stimulus, that is histamine via H2 receptor, but it blocks more than 90% secretion. We discuss all such types of interesting questions and clinical cases on our channel. So if you have not subscribed to the channel, it will be a good time to subscribe now, and also hit the bell icon.